Welcome everyone to the very first seminar in the new seminar series for the ARC Training Centre in Predictive Breeding. This series will be running seminars monthly, highlighting advances in predictive breeding science that's relevant to plant and animal breeding. So very exciting to kick things off today. But before I get too far, I'd like to acknowledge the traditional owners and their custodianship on the lands in which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. I'd like to introduce myself. I'm the director of the ARC Training Centre in Predictive Breeding, Professor Lee Hickey. It's my pleasure to be coordinating and helping bring people together through this centre across our 5 Australian university nodes. This centre is supported by the ARC, and we have nodes not only here at the University of Queensland, but also La Trobe University, UNE, University of Western Australia, and the University of Wollongong. So all coming together to help train the next generation of plant and animal breeders. Some housekeeping before we get underway. Everyone should be aware that this seminar is being recorded, so please hold your questions to the end for those of you here in person. For those of you who are joining online, thank you very much for tuning in. Please use the Q&A tab rather than the chat function. That way we can see and sort through those questions at the end for our speaker. So now I'd like to introduce our very first speaker for the seminar series, Professor Ben Hayes. He's a research theme leader within the training centre. He's also the research director for the CRC for Zero Net Emissions. Ben's a very busy man, taking on a lot. And I think I would say he's very much an innovator in genomics, predictive breeding science for plant and animal breeding. He's published more than 300 publications in the field. Probably a lot more. It's hard to keep track. He's a highly cited researcher, but the one thing that I really like about Ben, and I've had the pleasure of working with him for the past 9 years here at UQ, is that Ben continues to think big. And I think that's what we need to be doing to really advance our predictive breeding science for the future. And I hope that we can instill that kind of thinking in the next generation of breeders. So without any more words, I'd like to introduce Ben and hand over to him. Thanks very much, Ben. Please welcome Ben.
Okay, thank you very much, Lee. And I'll just start by saying how thrilled I am that this ARC Centre for Predictive Breeding is up and running. I think it's just this incredible opportunity for students to have serious resources for training, to build up your networks, and to have some really great discussions between you to come up with the new methodologies and technologies that we'll be using for breeding in the future. So I'd really encourage the students to take that opportunity. Leading into this talk, what I'm about to present really came out of coffee conversations, various dinner meetings, where a few of us got together and thought about where is breeding going to go in the next couple of decades. So I'd like to thank my co-authors up on the screen there. The ideas I'm about to present are really from the collective group there. Okay, so I'll start with genomic selection, and many of you know and I've done a lot of courses and work in genomic selection, but just to reiterate the main points, genomic selection starts with a very large reference population or a training set of individuals, plants or animals that are measured for your trait of interest, and they're also genotyped for genome-wide markers. We use those genome-wide markers to track the mutations that actually cause variation in that trait of interest. So using that reference population, we derive this prediction equation, which is the effect of the SNPs on the trait. Once you have that prediction equation, you can take your selection candidates, the individuals that are in line for breeding, and genotype them and then calculate their genomic breeding values. Once you have those, you can rank them and select the best individuals for breeding. So genomic selection's been implemented in many species, and as I'll show you, it's working pretty well in most cases. So in dairy cattle, genomic selection in Australian dairy cattle was implemented in 2011. And one of the traits we really focused on was fertility, because fertility in dairy cattle was actually going backwards quite quickly. So we put quite a lot of weight on fertility to try and improve that trait once we had genomic breeding values for fertility. So you can see from this graph with time on the x-axis and genetic level on the y-axis for fertility, that after about 2011, once we had these genomic breeding values available, the genetic trend for fertility has just gone up and up. So that's been quite a success story. In crops, a really standout example is the work done by Mark Cooper to develop corn hybrids that yield well under drought conditions. So these are called the Aquamax varieties, and they've really increased yield. And these varieties were developed with genomic breeding values. Okay, so if you think about what we have in our species where we've implemented genomic selection, we have a very large number of individuals with the trait data and with the genotypes. And I thought about livestock, and if you think about all the individuals that have been genotyped, pigs, cattle, chickens, you come up with something like 20 million animals around the world have genotypes. In crops, it would be many millions as well. So is there an alternative way you can use this data? Genomic selection's working well, but can we use the data somehow to accelerate our genetic gains? And what I'm about to present comes out of a paper we published in Nature Genetics at the end of last year. Okay, so in genomic selection, what you do, you calculate your genomic breeding value for your selection candidates based on their SNP genotypes, and you would select out the individual with the greatest breeding value to produce the next generation. In this case, it's line 1 there has the highest genomic breeding value. Okay, so the lines are ranked based on their genomic breeding value. But if you peer into the genomes of those individuals and you break the genome up into chunks and you start looking at their genomic breeding values, not for the entire individual, but for chunks of their genome, what you see is the good individuals have lots of good chunks, but they may occasionally carry a bad chunk, a chunk with a low breeding value, a bad haplotype, for example. Whereas an individual with a low genomic breeding value will be poor for many chunks across the genome, low local breeding values across the genome, but may have 1 or 2 really good chunks that you'd like to use in your breeding program. So in this alternative approach, which we've called optimum haplotype stacking, what you do is you look across the population to see which individuals are carrying the really good chunks of genome. And then you stack all those good chunks of genome together to create what we call the ultimate genotype. So the ultimate genotype is the best chunks of genome from right across the population, all stacked together. Now, those of you that have a long history in plant breeding, you'll be sitting there thinking, this sounds quite similar to gene pyramiding. And indeed, this concept of gene pyramiding, where you stack the best alleles at a few loci together into the one genotype, has been around for quite a long time. Ian tells me that the first occurrence of this in the literature is back in 1952, and that was a paper by Watson et al. This concept of gene pyramiding was developed further by Hospital more recently. But typically this gene pyramiding concept refers to a fairly small number of loci, perhaps 3 or 4. This idea of optimal haplotype stacking, it recognises the fact that for most of our complex traits, there are many mutations of small effect right across the genome that cause variation in that trait. So almost every chunk of chromosome that you look at will have some effect on the trait. If that is the case, you need to think about stacking all the best haplotypes in each chromosome segment across the genome into one individual. So just to go into detail a little bit more how that works, for each chunk, each chromosome block in the population, so each chromosome location, if you peer into that chromosome block for the population, there's going to be a range of haplotypes there. And the haplotypes are the unique combinations of SNP alleles that you observe in that chromosome chunk. And what those haplotypes are really doing is tracing back the chromosome segments that have come from the ancestors in the population. So for example, for this particular chromosome chunk, you've got several SNPs there. You look at the alleles and you see the first haplotype there is AT, CCCC, GTA. So that would be one of our haplotypes. And you can see 2 other haplotypes that exist at that chromosome block. You can work out the effect of those haplotypes by adding up the effects of the SNP alleles on that haplotype. And you can see here that the blue haplotype is the most favourable haplotype at that location. And you also have 2 undesirable haplotypes. So then you can look into your population and characterise for each individual, do they carry the most favourable haplotype at a location, or do they have undesirable haplotypes? And you can see here that Floss has the most desirable haplotypes across the genome, but she does have 2 locations where she has an undesirable haplotype. On the other hand, Axel has almost all undesirable haplotypes, but 1 desirable haplotype there. And what you want to do is stack up those desirable haplotypes into 1 ultimate individual. Just to show you what this looks like in real life, this is some work that Tong, our PhD student, did, and he was looking at rust resistance in wheat. So he had this reference population. He could work out the effect of the haplotypes across all the chromosome blocks in the genome, and then he could peer into each individual and see if they had a desirable or undesirable block. And what you see here is the wheat genome. Wheat is hexaploid. It actually has 3 genomes, A, B, and D. And what you have is the best individual is WLA221, the most resistant individual, and the least resistant individual is WLA091. In this case, the haplotypes with the most negative effect are actually desirable because they have the lowest rust scores. They're the most resistant. So what we see is that individual that is quite resistant, WLA221, has a stack of really red haplotypes, mostly red. That individual mostly carries desirable haplotypes. But if you look, you'll see there are just a few undesirable haplotypes in there. If you look at the really susceptible line, WLA 091, you can see most of its haplotypes are blue, undesirable, more susceptible, but there's just a few locations where it has quite desirable haplotypes in red. So that's just an example of what these desirable and undesirable haplotypes look like in a real individuals. Okay, now the interesting part comes if you look at the performance of these ultimate individuals or the predictive performance. So if you stack up all these desirable haplotypes into one individual, you get an individual that has a predictive performance many times better than the current best line. So in dairy cattle, when people have done this, the performance is 6 times the current best performing bull in the population. So both Cole and Van Reyden and Kemper did this on an index of the traits that affect profit for dairy cattle. So it's milk production, fertility, survival, and so on. And they came up with individuals that were 6 times better than the best performing individual in the population today. For yield, Vosfels did this. It was a little bit lower increase in performance, perhaps reflecting the fact that wheat is a bit less diverse than dairy cattle. It's a little bit more inbred. For rust resistance, Tong came up with this figure of the best line you could create, the ultimate genotype, was twice as resistant as the best existing line. And we also did this in chickpea, and you come up with these ultimate genotypes that have a 5-fold better performance than the best current line. So that's pretty astounding, and we'd really like to breed lines with that level of performance. So the next question is, how do we get there? So how rapidly can we stack those desirable haplotypes and get to this ultimate genotype? And we did some thinking on this, and it turns out the time taken to stack all these most desirable haplotypes into a single genotype depends on the size of your chromosome blocks. Obviously, if you have really big blocks, that's easier to stack than lots of little blocks. The number of haplotypes within each block, the effective population size, that's really important. The smaller your effective population size, the more inbred, the less different haplotypes you're going to have to stack. The number of crosses per generation, the more you have, the more recombination there is, the more you can break up these chromosomes to get your ultimate desirable haplotypes coming together. And then the number of progeny per cross. That's really important to, if you think about a pair of parents that contain desirable and undesirable haplotypes, to get those recombinations so you can get those desirable haplotypes on the one chromosome. And finally, the recombination rate tells you how likely you are to get a recombination between a desirable and undesirable block. So just looking at what people have thought about for block size, this paper by Cole and Van Reyden, which was the first one to think about this stacking to build ultimate genotypes, they just considered entire chromosomes. So they looked in the US dairy bull population, and they said, if we just take the best chromosome in the population and we put it together, what ultimate genotype can we build? And it turns out the net merit of that ultimate genotype, so the dollar value, was about $7,000, a little bit higher. The best current bull in the US population is about $2,500. That's based on the value of its genes for milk production, fertility, disease, and so on. So that's pretty impressive, but that's whole chromosome blocks. There's going to be lots of mutations affecting the traits on each chromosome. So really, you'd like to look at smaller blocks so you can get recombination between desirable mutations and the undesirable ones, so you can stack the most desirable ones. And typically, when we run simulations now, and I'll show you some of those in a moment, we think about stacking anything from a quarter of a chromosome down to quite small blocks, 100 kilobases. And I'll show you examples of that. Okay, so other parameters affecting how quickly you get to the ultimate genotype, number of crosses per generation, and number of progeny per cross. Before I go into that, I just want to point out that selecting the group of parents that between them contain all the ultimate haplotypes out of your whole population is actually a really challenging problem. It's because there's so many possible combinations of selecting, say, 25 parents out of 100 possible parents, that it's an enormous number of possibilities to sort through to find the optimum group of parents. And in fact, if you have 25 crosses that you want to make, and there's 100 possible parents, the number of possible crosses is 8 by 10 to the power 51. That is a huge number. So you need a really nice optimization strategy to choose that best group of parents. In our work, we use a genetic algorithm to do this. Some people call a GA a type of artificial intelligence, which it is, but it was actually developed back in the 70s, inspired by genetics, having chromosomes that recombine and mutate. And those concepts are used to generate solutions for optimization problems. In this case, we code up, is a parent going to be chosen for mating or not as a gene? And you might have 25 genes across your chromosome. And you run it through the GA, and it gives you eventually your best group of parents. Okay, so that's how we optimize this problem. To get back to those parameters of number of crosses per generation and number of progeny per generation, and how does that affect the speed at which you can get to the ultimate genotype? So one of our now students, Kira, has spent quite a lot of time on this, done a lot of really nice simulation work to investigate these parameters. So the simulations Kira ran started with a real wheat population. It's a wheat NAM, for those of you that know it. It's got 5,000 markers, 50 founder lines. And the problem was to select out of those lines, or the 50 founder lines, the lines that contain all the most desirable chromosome haplotypes, sorry. Okay, so the problem was select those founder genotypes. From those founder genotypes, a number of parents for crossing, cross them, produce a number of progeny, and then repeat that process over and over again to see how close you could get to that ultimate genotype. These are the results, so I'll take you through these carefully. So in this simulation, there was either one progeny per cross. This is just an example with 25 crosses. There was one progeny per cross, 10 progeny per cross, or 100 progeny per cross. The strategies we investigated was using the genetic algorithm in this optimum haplotype selection to choose the parents in each generation. And we compared that to what people do now which is just selecting on genomic breeding value. So ranking the candidates, selecting the best ones to breed from. The other strategy that we tried, or Kira tried to be precise, is starting out with that genetic algorithm. So choosing the group of parents that between them contained all the desirable haplotypes, and then running truncation selection after that. So if you look at the results, the first graph there, generation, and the genomic breeding value on the y-axis. So how much genetic gain do you make? The interesting thing is truncation selection actually does really well in the first generations. So that's the orange and yellow lines there. But truncation selection runs out of variation to use fairly quickly in these simulations. And after a certain number of generations, the optimum haplotype selection starts to beat it. So that comes in about 70 generations in the future. So you can get greater genetic gain with this strategy. But that, if you think about it, that's quite a long way in the future. With speed breeding that Lee and colleagues have developed, you might be able to get through 5 or 6 generations a year. So that could be 15 years of breeding, for example, but it's still quite a long time. Turns out the strategy that does the best is this hybrid. So starting with a population that contains all the desirable haplotypes and then just letting truncation selection rip. That really does quite well. The other interesting thing that I'll come back to is the amount of diversity retained in the population. And that's that second graph there. You can see that truncation selection very quickly runs out of diversity, whereas the optimum haplotype selection retains quite a lot of diversity. Why is that? It's because in the optimal haplotype selection, it's choosing individuals that have maybe just one really good haplotype and the rest not so good for that trait. So it's pulling in a lot of diverse haplotypes. Okay, so that's the theory. What about in practice? So we're very fortunate to have had a project with long-reach plant breeders to put this idea into practice. And we call this fast stack because it's this idea of optimal haplotype selection combined with speed breeding to run the generations really quickly. So over the past 5 years, probably now, Eric and Delaney and others in the group have been implementing this strategy in the glasshouse using long-reach material, germplasm, as a starting point. And it's been really quite exciting. And I should say what they implemented was that hybrid scheme that I put up before. It's been really quite exciting because about a year ago, we were up to the point of putting this material, the fast stack material, into the field and looking at how it performed compared to the current best varieties. So Eric put this figure together, and what you see there is the field trial sites at different regions in Australia, and the blue bars are for these fast stacked varieties, whereas the red is for the check varieties, and the y-axis there is yield. So our fast stack lines in almost every environment, I think it is in every environment, are performing better than the current checks. So it's really quite exciting. I would say this is 1 year of field trial data. We need more years to just verify that this works across the variation in years and so on, but it's looking really promising. And some of these lines are now being taken through the release pipeline, I believe. Okay, that's in crops. We've got an example of where it's worked very well. We don't have any example in livestock as yet where there's calves or little chickens or little pigs on the ground yet. What we really need in livestock is a way to speed up the generation interval, reduce that generation interval down to months instead of 4 years like it is in cattle. So in cattle and sheep, for example, you can breed larger numbers of offspring per cross by using things like in vitro fertilization. So you can get more recombination to start to stack up those desirable haplotypes. But from IVF, you typically get between 5, if it's working really, really well, maybe 10 offspring per mating. It's still not enough to get where we want to go. Another technology that is being developed is in vitro meiosis. And this is basically getting recombination in the test tube. So what you do in vitro meiosis is you take stem cells, might be from an embryo, and you push those stem cells to form oocytes, so down the female germline, or sperm. And you have the oocytes and the sperm in the dish, you put them together, and you have meiosis, fertilization as well, and then you create little new embryos and then do the whole thing again. And by doing that, you can get multiple rounds of meiosis really quite quickly. This sounds pretty science fiction. They've actually done it for the female side of things in mice. They have produced little oocytes in the test tube from stem cells. Then they've fertilized those and produced little fetuses in the test tube. So it's a technology that is coming along, but it's not quite there yet. Okay, aquaculture that I know some of you work in. I think aquaculture is actually a place where this idea could be quite readily applied. And that's because from a cross, male and female, you can get millions of offspring. So the chance that you get the recombinations to stack the best haplotypes in the parents is really quite high. You've got the issue of cost of genotyping, but aside from that, I think aquaculture would be a great place to try this. Okay, so now for the second part of this talk, we're going to diverge from reality, what's almost practical now, and start thinking about what we could do in, say, 20 years' time. So this is looking at what technologies are being developed, coming up, and applying some of those technologies to this problem. So I'm going to go through 3. One is accelerated and guided recombination. The other is direct editing of ultimate sequence haplotypes. And the last one is writing out the ultimate genotype, the DNA, directly. So accelerated and guiding recombination, about once every decade, somebody rediscovers that recombination is actually heritable and gets excited about that again. But if you look back, that's actually been known since the 60s, I think. People working in fruit flies created lines that were high and low for recombination, which is pretty amazing. More recently, various groups have been using CRISPR-Cas9 to induce recombinations at specific sites in the genome. If you think about that in relation to our stacking problem to build the ultimate genotype, it is potentially pretty exciting. You could have a recombination and stack 2 desirable haplotypes really quite quickly. One thing to point out is just doing this randomly along the genome, just having a boosted rate of recombination. When people do realistic simulations of this, it doesn't seem to be really beneficial, because what happens is those SNP allele mutation allele, so the mutations that are really affecting the trait, those associations get broken down by the recombination. So those SNP effects you thought you had were predicting the mutation effect. They actually start breaking down as you boost the rate of recombination. So this paper here, if it ain't broke, don't fix it, evaluating the effect of increasing recombination, pointed that out. I think what is going to be more valuable is targeted recombination. So if you can target recombination between the blocks, that could increase your speed with which you get to the ultimate genotype. And Kira, again, has done simulations to look at this. So on this graph, we have, again, generations and genetic gain on the y-axis. And you can see there's new lines now. For example, the dark blue up the top there is this hybrid scheme where you have a higher rate of recombination. And in fact, you do get accelerated gains and a higher level of ultimate gain as well. So that's exciting, good avenue to explore. The simulations say it's still going to take a lot of generations to get there though. So can we do something else? What about using our gene editing technology to just write in the sequence of our most desirable haplotypes? So when people think of gene editing, they think of targeting a specific mutation because they know that mutation has a big effect. For complex traits like yield in crops, yield in dairy cattle, it's actually been really difficult to find these mutations. We have very few examples of mutations that are known that affect these complex traits. What we do know now, though, from all our genomic data, is the sequence of haplotypes that somewhere within that haplotype contains that mutation. So there might be 20 or so mutations in there. We know that one of them affects the trait. So what we could do is just go in and write that desirable haplotype sequence, knowing that we're editing the desirable mutation, but we're also editing 9 mutations that don't do anything. So with our large volumes of sequence data available, this could actually be possible. What it does depend on, though, is systems where you can make large numbers of edits simultaneously. I think the best that anyone's done so far is about 25 edits at once, unless there's been a new paper published in the past few weeks. There's a bit of a race to do this, but it's about 25. So let's just have a quick look how many edits we need to make to do this. So Christy Warburton, another one of our scientists, took haplotypes from our cattle population, so sequence haplotypes from the 1,000 Bull Genomes Project, and looked how many edits you would need to make in a 100 kilobase haplotype to get from a just average haplotype to the most desirable haplotype. If you are just considering haplotypes within a breed, to go from average to best, you had to make about 142 edits. That's quite a lot. And that's just for 100 kilobase chromosome block. Between subspecies, say you wanted to edit in really good adaptation, heat tolerance from Brahmins into Angus, much more widely separated, those two populations, you're looking at something like 400 edits you have to make. That's a lot. So at the moment, that's not feasible. In the future, it may be as people develop more and more technologies to get these simultaneous edits. Okay, another strategy that Tim Marnie actually came up with uses, I'm not sure if I should call it an old technology or a technology that's been around for a while, but is tried and tested, which is to create these yeast artificial chromosomes with fragments of, in this case, cattle genome inserted into those yeast artificial chromosomes. People used to do this to create physical maps of the genome and for all kinds of other reasons. But what you could do is clone in a desirable haplotype into your yeast artificial chromosome, create a collection of those that contained all the most desirable haplotypes across the genome, and then insert those chunks, the entire chunk, into the genome of your target animal to start building up those desirable haplotypes. And that is probably more feasible than doing the many thousands of edits that we need to get to the ultimate genotype. So that would be a very interesting strategy to start thinking about. Just finally, before I go on, the advantage of doing that is you've actually seen those haplotypes in living animals. If you do gene editing of a particular base that you think might affect your trait, if you've never seen that before in your population, there's no examples of that, you might actually be creating recessive defects. And particularly if you're doing a large number of those, you could create a recessive defect. So you definitely don't want to do that. The advantage of these writing in these desirable haplotypes or cloning them in is you've actually seen them in living animals. So they're compatible with life or living plants. If you were going to be really cautious, you'd make sure you'd seen it in the homozygous state. So 2 of those haplotypes together. So you knew there weren't recessive defects there. Okay, so moving on from that, the last strategy, and something that may become possible in the next few years for some of our species, is just writing in the whole genome of the species. So you've got your sequence of your ultimate genotype, you take that and you just print out the genome and create these individuals directly with this ultimate genotype. So this synthetic biology approach is perhaps a lot closer than you think. In yeast, there was this publication that came out last year where for half the yeast genome, they've actually written out this genome, printed out the DNA and inserted it into a yeast cell and recovered a functioning yeast organism. So that's really quite incredible. At least in yeast, it's a long way down the track. A Chinese group has done something similar, though not half the genome, rather a small chunk of the genome in moss. And people are trying it in other species as well. So as long as we have this ultimate genotype, the sequence of the ultimate genotype, at least we know what we want to write when we do this synthetic biology approach. So that's something to keep an eye on as well. Okay, so to finish off now, these ultimate genotypes, they look very attractive because they can have performance up to 6-fold greater than the current best individuals in our population. And this approach is really marrying up complex traits and gene editing synthetic biology. There's always been a bit of a schism between complex traits and something like gene editing, because gene editing looks for 1 or 2 locations in the genome that you could edit. This approach really is a way of using gene editing or synthetic biology to improve our complex traits, not just Mendelian traits, for example. But how realistic is it to think about getting 6-fold increase in performance? So, in fact, that's already happened. In dairy cattle, for example, if you compare milk production from 1940 to the current day, milk production has increased about 5 times from cattle. So it's not that different from 6-fold. In maize, there's been this beautiful long-term selection experiment for increased oil in the kernel. And you can see that's generation time, the amount of oil in the kernel. The selection response just goes up and up and up. And you've got many-fold increase over where the population started. I think a really key thing though, or it's going to be really important, that you don't just select for one trait. History says that's a bad idea. For example, in dairy cattle, when people did that, they got a really strong negative response for fertility because there was a pleiotropic, lots of pleiotropic effects, and there are lots of examples of that. So it's going to be really important when you think about an ultimate genotype to think about that genotype for multiple traits, including fitness, how well the plant or animal functions. So traits like fertility and disease resistance. The other thing to keep in mind, it's a bit, what would you say, egotistical or hubristic to think we know the ultimate genotype for a species. That's going to change over time. We've got climate change. We're going to need more and more heat tolerance, resistance to different diseases. Consumer preferences for food is changing all the time. So that ultimate genotype that you're targeting, it's actually a shifting target. But that reinforces what we need is methods to get there really quickly. We also need, a really important point, is we need to maintain diversity in our populations so that we do have the flexibility to shift our target. We've still got genetic variation that allows us to go this direction or that direction. And just to point out that as people have implemented genomic selection, what they've seen is quite an increase in the inbreeding in those populations. So this graph just shows the inbreeding in the Canadian dairy population in different decades that you've got, the rate of inbreeding. If you just look at the green bars there, that's one measure of inbreeding. And you can see in this last decade, which is the decade where genomic selection was implemented, the inbreeding has really accelerated. And this is because what actually happens in the dairy breeding programs, these young bulls get tested, get genotyped, they come up with amazing genomic breeding values, spectacular. Every breeding company wants either that bull or offspring from that bull in its cohort of artificial insemination sires, and the population really narrows down very quickly. It's slightly frustrating because the geneticists have come up with ways of managing inbreeding, but I think the lure of these really high-performing bulls has just overridden this knowledge that you have to control inbreeding. So that's resulted in this kick up. So we really do have to watch that. And the nice thing about our optimum haplotype selection and this fast stack approach is that it can actually maintain diversity because you're dragging along those individuals that might just have one desirable haplotype and the rest of their background is quite different to the other individuals. You do maintain some diversity. Okay, but these potential negative effects are something we really have to watch. And there's a paper published just recently about the negative impact of genomic selection. That's really because we need to put more emphasis on fitness, disease resistance, and so on. And I'll leave it there, but just a huge thank you to everybody whose ideas and work and effort went into what you saw today. Over to you, Luke.
Thanks so much, Ben. And thanks everyone for tuning in online. Thanks for the great attendance here in person. We're going to open it up to for questions now. If you're online, please post your question in the Q&A function, but we might to get things going, might take some questions from the floor here, from those in person. And we might just ask Ben to repeat the question in your response.
So Ben, thanks for a great seminar. You had a value function there, but you could equally have a cost function, right? Because if you're a plant breeding company, not all traits would be worth taking to the right-hand side there. So can we optimize on that basis as well to make the most commercial progress?
Yeah, I think that's a really good point. And Tong, our PhD student, actually came up with a similar idea to what you're articulating. So, sorry, to repeat Matthew's question, it was for every trait, we may not need to get to the ultimate genotype. It may be sufficient just to have a certain number of desirable haplotypes. And a good example of that is rust resistance in wheat. You may only need 10 desirable haplotypes to be completely resistant. And in fact, we're looking at strategies to take that into account. Yep.
Okay, we have another question.
Hi, thanks for the lecture. I just have a question about the genomic blocks that this all revolves around. The way that it was presented here, it seemed to assume that all of the blocks are compatible very neatly across with each other. And my experience with genomics has told me that something that neat is almost never the case. Is this a technical issue worth worrying about, or am I seeing shadows?
No, you're quite right. If you build the blocks based on LD, linkage disequilibrium, you get very different physical sizes of those blocks, reflecting really the recombination, or ultimately the recombination hotspots. So one approach we take in real life is to build these blocks based on the LD patterns that we see. So in some regions, you'll have quite small blocks because there's a lot of recombination, so a lot of haplotypes. In other regions, there's very little recombination. So the physical length of the block is quite big. Yep. So in real life, it's variable. They were just cartoons to get the point across, I guess.
Yes, but that leads me to, doesn't that have a further problem whereby you'll have a haplotype that you want and a haplotype that you definitely don't want, and they'll just come as a package deal?
Yes, if you've got an individual that has a very desirable haplotype right beside an undesirable haplotype, the worst is if it's nested within two very undesirable haplotypes, you need a double recombinant to release that haplotype into a different background. And the probability of that is quite small. So one way of overcoming that is just to generate lots and lots of progeny. The other way of doing it is targeted recombination with your CRISPR-Cas9. But yes, it's a constraint. Yep.
All right, thanks. Let's hope that synthetic biology comes along.
Yeah.
Ben, I actually have a question. I thought it might be interesting to expand a bit on some of the ideas you have around epistasis.
Oh, yes.
And so what are the opportunities to consider epistasis in some of the estimating the ultimate genotype and how we get there?
Yep. So one of our scientists, Seema Yadav, is actually working on this at the moment. So you can imagine that if you have large epistatic interactions, what you want to do is think about not just stacking the blocks because of their additive effects, but combinations of blocks that, when you consider the interactions, give you a higher performing ultimate genotype. The challenge is searching through all the parents to take advantage of those epistatic interactions becomes just an enormous problem, larger than the one I put up for the genetic algorithm. But we are trying to find optimization procedures that will be able to deal with that. But it's challenging. Yep.
Thanks for the talk, Ben.
you mentioned a bit about multi-trait selection. And I guess traditionally you would index GBVs across multiple traits kind of approach. And that's a sort of summation of the issue. Now that if you're breaking things down into these blocks, what's your ideas on how you'd account for the fact that some blocks will push things forward and others will pull things back on a per trait basis? What's your thoughts on how you'd go about actually doing that selection of blocks in the first place once you're considering multiple traits that have that push and pull effect? Yeah, that's a really good question and one we're trying to solve at the moment. So another way of framing the question is, if you have multiple traits, do you get to the same endpoint if you just stack on the selection index? So you calculate the GBV for the selection index for each haplotype, and you just stack the haplotypes to get the greatest sum of all those haplotypes. Or can you do something better? Can you come up with some strategy that gets you to the ultimate genotype that has the highest selection index value that is better than just considering the selection index for each haplotype? And I'm not sure what the answer is, but we're certainly going to try using some of these AI methods to ask that question. Yep. Come up with breeding strategies that give you the best multi-trait ultimate genotype. Yep.
Okay, we've just got an interesting question here in the Q&A online, Ben. So from Professor Graham Hammer, the ultimate genotype will differ every year in field crops. How are you going to deal with G by E? Maybe you'd like to elaborate on some of the strategies.
Yeah, really good question, Graham. Thank you. This is something we're actively thinking about. We haven't solved it either. Of course, with your haplotypes, you can consider just not the average effect, but the G by E effects for those haplotypes. So work out which haplotypes performs best in each environment. And then you come to your breeding strategy. What do you actually do with this? Do you create an ultimate genotype for every different environment, which is going to take an enormous amount of breeding program resources? Or do you have just a couple of target environments and calculate kind of an average ultimate genotype that performs best in that those broad environments? So that's a really good question. We're looking at it now, and it's really going to depend on the magnitude of the G by E. If the two environments have a correlation for performance of 0.8, it's probably not worth splitting your breeding program into two and breeding for the two separate environments. If it's 0.5 though, it almost certainly is.
Some good rules of thumbs there. I've just got before we take a question here in person, we've got a comment here, which is pretty interesting, I thought. So all of the yeast chromosomes have now been created synthetically. Macquarie Uni completed the last chromosome and published it. There are now several groups wanting to fully synthesize the moss genome. Pretty cool.
Excellent.
So, question at the back there.
Yeah, I have a question. Thanks, Ben, for your excellent talk. If you look traditionally, you had a breeding value for each individual in a population. But to get your ultimate genotype to stack it, you may actually look at it differently by having breeding values for those blocks, isn't it? Because you really are not interested. You're not getting the most effective approach by starting with the best individuals if you want certain combinations at the end. Considering starting values for each block may not be in the most elite parents for start. Point?
Yes, that's pretty much what we do. So I'll just repeat Andre's question. So wouldn't we be better off having breeding values for each block and then looking across the whole population to get the individuals that had the best collections of those we call them local genomic breeding values. And that's pretty close to what we do. In a real plant breeding program, the question is, do you start with just your elite germplasm, or do you go much wider than that? And I think we're starting to think that it'll be better to go quite a bit wider than that. And for example, for disease, you have your elite population, but you also have these germplasm collections. And now we're calculating those local GBVs, haplotype effects, on those germplasm collections as well. And we're thinking about strategies to combine the elite germplasm and the individuals that carry the best local GBV from those germplasm collections. Yep.
Okay, we might have to stop there. We'll please thank Ben again for a great talk. If you're interested in knowing what lies ahead around the seminars, please check out the website, and you can also sign up to our newsletter as part of the ARC Training Centre in Predictive Breeding.